Good evening and welcome to this Absent seminar. I'm Barry Smith, the Director of the Centre for the Study of the Senses at the University of London, and I'm standing in for Chris Kelly tonight, who's indisposed, but she will be back with us soon and we wish her well. So the seminar this evening is on the topic of parosmia, which I know a lot of people have been interested in. Some people have been undergoing some of the symptoms. And so this seminar is an attempt to bring together scientists and clinicians who are going to look at what we know so far and what the latest research can tell us. And I'm delighted to be joined this evening by Dr. Jane Parker, who's an associate professor of the Department of Flavor Chemistry at the University of Reading. And Jane has worked as a sensory scientist on flavor analysis and on the underlying chemistry of the flavors in our foods and drinks. And before the COVID-19 outbreak, she had already begun a very interesting project on anosmia, seeking to identify some of the molecules that might trigger the smells that people suffer from when they have parosmia. We're also joined, I'm glad to say, by a regular on the Absent webinar series, and that's Dr. Simon Gaine. And Simon is an ENT consultant at the Royal National Throat, Nose and Ear Hospital. Simon's also a long-term researcher on olfaction and rhinology. And I'm very glad to say that both Jane and Simon are fellows of the Institute of Philosophy's Center for Olfactory Research and Application. So these are colleagues of mine, and I'm delighted to have them with us. Now,
Nice to be here.
Nice to have you. Nice to see you both. Now, the topic of this evening's seminar, of course, is familiar to a lot of people, but I thought I would start just by bringing to everyone's attention, because it may chime in with something that you yourself have experienced, to bring out the topic of how much of an impact the nature of parosmia has, this idea of familiar things being distorted in the way they smell to us, and how much of an impact that has on people's lives, their health, and their well-being. So here's a quote from the Facebook group on loss of smell and taste that Absent have been running. It is so difficult to describe it to people that haven't experienced it. It's been hard for people even close to me to understand the severity of the loss and how it affected my life. And another quote that struck me very forcefully, someone who hasn't experienced this, and this was from a Twitter post. I thought having anosmia was bad, but parosmia is horrendous. Food smells and tastes like fruity sewage, constantly battling with nausea and no desire to eat. So this gives us a sense of just how severe this condition is, and we're going to talk about it in a moment. But I would also like to welcome Aidan Kirkwood, who's going to look after the technical side of things for us this evening, and also field questions. And please do put into the Q&A questions when you have them. We'll try to take some as we go. We may not get around to answering all the questions, but we'll do what we can, and Aidan's going to let me have word of those when they come up. So I thought, Simon, I would come to you first, if I may, because I guess we're seeing a lot more cases of parosmia, and I wondered how that's striking you as a clinician, what way people are presenting to you with this condition. And it's true to say, isn't it, that this isn't a new condition that's come with COVID-19. It's been around for a while, hasn't it?
Yeah, I mean, parosmia has been described for almost as long as people have been talking about smell disorders, and that's why when we're talking now about what changes in the sense of smell happen after a viral infection or anything else, we've gone away from using anosmia or hyposmia as a sort of catch-all to talking about olfactory dysfunction. So although the sensitivity of your sense of smell is one axis on which your smell function can vary, so you're better or worse at smelling things, and that's what we call normosmia for normal smell function. hyposmia for poor smell function and anosmia for no smell function, we've realized that there are other axes on which your smell can go wrong, even though you may be able to detect things. And that's where the other olfactory dysfunctions come along. Parosmia is probably the most prevalent of this one, and it's also called troposmia. And I've always liked troposmia as a better term for it, but unfortunately it's not the common term. But it was when these things first started being investigated, it was proposed as another word for it. And tropos is to twist, so it's a twisting of the sense of smell, and that gives an idea of the kind of things that we look for in trying to make the diagnosis of a parosmia. So this condition is triggered, number one. So it is a response to an environmental stimulus in the way that other conditions such as phantosmia or olfactory hallucination aren't. They just appear out of nowhere with no obvious chemosensory stimulus. So it's triggered. It's fairly time limited in that the smell tends to have roughly the same amount of take the same amount of time. The sensation takes the same amount of time as the original smell, but it is perceived as distorted. It's not the smell that you're expecting, and it may be that conflict between the smell you're expecting and the smell you're getting that is part of what makes it so unpleasant. And that unpleasantness, that negative hedonic value, is almost universal. And the fourth characteristic of parosmia, when we talk about it, so unpleasant, triggered by something in environment, doesn't last for too long, but it is distorted, and sometimes the original character of the smell you're smelling comes through a little bit, but it's usually described in ways that try to evoke this feeling of disgust. And disgust is very closely associated with a lot of smells for us, because smell is a protective sense that you want to have some hardwired avoidant behaviors in smells in organisms, because you don't want them eating or drinking things that are unpleasant to them. So when we talk about parosmia, those are the kind of things that we're talking about. It seems to see something gets scrambled. And you're absolutely right. We've seen it for a long period of time. It's characteristic of what we call neural smell disorders. So things that affect the first nerve of smelling, and that's really post-infectious, what used to be called post-viral olfactory dysfunction, and traumatic brain injury as well. So head injuries can do it as well. But it's really come, as a lot of smell disorders have come to the forefront in the last year with COVID, because COVID seems to be amazingly powerful at altering chemosensation in ways that we still don't fully understand. I think it's given us a very unpleasantly rich set of syndromes to deal with, and parosmia seems to be, it's difficult to say whether it's more common in the patients who've lost their sense of smell from COVID than those who lost their sense of smell from another virus, but it's certainly, because everybody's losing their sense of smell, it's much more common now. And it's for some people, very distressing phenomenon.
Is it true to say that there's a pattern of its appearance this time? I mean, are people first having anosmia, recovering the sense of smell, and then developing parosmia?
Yeah, that's the typical one we're seeing. Again, you can see it start from there's sort of varying stories, but the typical one that my patients are telling me almost over and over again is exactly what you're saying: sudden onset of anosmia, complete loss of smell, and for the majority of those people, they get better. If you look at the general population, 80%, 90% get better within 2 to 4 weeks. Their sense of smell comes back as good as it was before, or maybe slightly less, and that's it. And they just carry on with their lives. But the ones that the common description is, I thought I was getting better, my sense of smell returned, and suddenly, a couple of days after that, everything started smelling rotten. And that rotten sewage, dead flower water, those kind of metallic, unpleasant smells. These are the kind of qualities that people ascribe to the Porosmia smell. And
of course, exactly as you said, that the nose is a kind of gatekeeper for what we will allow to take inside us and think acceptable. So if you're getting those kind of smells, foods that you would otherwise quite like and enjoy, they're going to be repellent. And even if you know that it's distorted, it's probably very difficult to overcome your own response to this.
Yeah, I mean, this is part of, as we talked about before, this is the interesting part about smell is how deeply wired it is into bits of our brain that aren't necessarily very conscious. So the areas that have to do with instinct, with not learned behavior, but essentially programmed behavior. And so that overwhelming sensation that this is something that is disgusting and should be avoided, you're not going to overcome millions of years of evolutionary programming just by thinking that a banana tastes nice, when every sense, the most important sense that communicates disgust, we don't get disgust from hearing and sight particularly. We get it from smell and taste. So that sense of telling you, no, this is off, this is disgusting, this is unhealthy, this is bad, it's very difficult to overcome that.
And so I want to turn to Jane, because I want us to talk about the research you've been doing, but maybe first, before we do, just to say what seems to happen in a lot of cases is that things that people quite like and have treated as familiar and enjoyable seem to be the targets of this experience. And so you've started to look at what those are, and as a flavor chemist, you've been interested in finding out, is there anything particular about those foods and beverages that are causing this? So maybe you can talk a little bit about it targeting particular things, and then I'm going to invite you to say more about how you've conducted the research.
Okay, so as you were alluding to, Barry, there's two things we've been looking at, and one is food triggers. What are the foods, drinks, or other items that trigger parosmia? But as a flavor chemist, I'm really interested in much more detail in molecular triggers. So I'll be talking about food triggers, but also molecular triggers. So are there molecules in the food that actually trigger this parosmia, or is it just everything in that food that just triggers parosmia? Is it specific triggers? And having looked at the list of foods, the preliminary list of foods that Chrissy drew up, some 18 months ago, there was coffee, there was meat, there was cucumber. And as a flavor chemist, I could identify the sorts of compounds that might be triggers just because they're so potent. They're really potent. You can detect, an average person can detect these potent aroma compounds at 1 part in 10 to the 14, which is just a tiny amount. So our hypothesis was that these were the food triggers. We've got these were the parosmia triggers, molecular triggers. So that's where it all started, and that's how I, as a chemist, became involved, because I understood the flavor chemistry. I wanted to apply it to this problem.
So is it's true to say, is it that when we react to these foods and drinks that we've liked, like coffee or roast meat and so on, and we now find them disgusting, they always had some of those kind of whiffy chemicals in them, I guess. I mean, coffee's got 1,000 or so volatile molecules, so it must have some nasty things in it. So are there any thoughts about why we don't usually have these reactions to these compounds?
Well, these nasty things, some of them are sulfur compounds, and it's all to do with the dose as well, because, at very low levels, these things are pleasant. But as the concentration increases of these molecules, that's when they start to get really, really revolting. But we need to distinguish between what's revolting and what's parosmic, because they're not the same. Because for normosmics, these sulfur compounds are really quite pungent. You can describe them with some, we can describe them as disgusting, strong, sweaty, all the rest of it, but they're not triggering parosmia. But if you dilute them far enough, there's a lot of sulfur compounds that give you lovely fruity notes, lovely floral notes, lovely meaty notes. So that's part of it. But the other part is that amongst these 1,000 other volatiles that are in coffee, perhaps 100 or so of them actually smell. And some of them smell very pleasant. And what they do is round out or balance out the whole flavour of the coffee. So we've got these sulfur compounds in there at low levels, so they're not too obnoxious. And we've got all the supporting cast as well. So we've got this rounded coffee flavour. And I think what's happening in Porosmia is that these sulfur compounds are poking through, and we're not getting the other compounds that actually round out the flavour. Or that's part of the story.
So I want to look at how you found that out, but I want to back up a little and say you had already been interested in people's odor responses to familiar foods and the distortions before COVID came along. So this must have now provided you with even more people interested. The press are interested, you know, the attention. We all had to learn the word anosmia if we hadn't already known it. Now people are having to learn the word parosmia. So you've been recruiting participants, I know, and maybe you could tell us something about what you do with your participants.
Well, okay, so the first batch of participants we recruited were all pre-COVID, and we got 15 of those, and we got those in between October and March. So October last year is when we started, and March. And we do a couple of things. The first thing we do is assess them for their olfactory function. So we use a set of sniffing sticks, which is a recognised medical diagnostic kit, which determines how good your olfactory function is. And from that, we'll get a score. There are 3 parts to it. I'm sure Chrissy's talked about it before, but there's a threshold part where we see how far we can dilute something before you lose the smell. There's a discrimination part where you have to see if you can pick the odd one out of 3 aromas. And then there's an identification part where you actually have to identify what's there. So we've got the scores. The first thing we do is assess the volunteers for their olfactory function. And then we sit them down in front of our GC olfactometer. And that's an instrument that can separate out these hundreds and thousands of volatile compounds in the coffee. So there's a long column inside it. It's a capillary, and we put the coffee aroma in one end, and the compounds travel around the column. Some go quickly, some go slowly, and they're separated. So the ones that go quickly come out first. And then we sit the volunteer in front of a little nose piece, a little glass nose piece that just fits on your nose. And as these compounds come out, we ask them to give us 3 bits of information. So what does it smell like, if you can describe it? How strong is it? And is it porosmic? And yeah, I mean, the first thing with describing it is most people find it incredibly hard to describe things coming off the GCO, even without Porosmia. So sure. if you put a new person in front of a GCO with these smells just coming, there's no cues, there's no labels. It might be orange, but there's nothing orange there to tell you what it is. So it's a hard job anyway. But when you've got Porosmia, it's even harder, because when these porosmic smells come out, they're smells that so many people say they just can't describe them. They're out of this world. They've not experienced them before. And you can't describe something. You always describe aroma by
something familiar, yeah,
relating it to something familiar. And if it's just nothing familiar out there, so people try and often use sewage, chemical, sweet, sickly sweet. Bin juice is one that crops up quite a lot, but they're all disgusting terms. So they've got to describe them. And most people resort to saying, okay, that's porosmic. It's that porosmic smell. Or some people say that's new coffee, because we had old coffee, as in what coffee used to smell like, and then new coffee that's coffee, but it's what coffee smells like now.
Well, that's really that's very helpful, because what they're doing for you is not just saying, because it must be a very unusual circumstance to smell all the component parts separately of a mixture. I mean, already it's weird. You don't usually do that.
Yeah.
It's very helpful if they can say that, oh, there's that nasty porosmic So that gets you a label or a tag for what they're smelling that they recognize as that. I guess that's the breakthrough there.
What's even more interesting, we've had some people say, okay, I've got 3 different porosmic smells, and they can tell me it's number 1, it's number 2, or it's number 3. And the ones that's number 1, for example, is they say, well, that's chemical, sewage, rubbery, smoky, cigarette smell. Those are terms that often come up. So there's one that's quite cigarette-y, smoky. And then there's this other one that's sort of sickly sweet, rotting type smell. And then there's a third one. I had one person who had 3 very distinct smells, and the third one was the onion garlic smell, which he didn't get in coffee, but he got when he moved on to onion. So even we can now split them out into different, different Porosmia smells. And I think that it's a sign of recovery. I'm not sure if I'm right, but I think when you start off with Porosmia, everything just smells like Porosmia. And very slowly, I think you then start to separate them out. You can discriminate between groups of compounds. And then as you continue to recover, I think that's when your normal sense of smell starts to come back. But I just found it fascinating that we're 3 different porosmic perceptions or smells. And I could actually relate these to different chemical classes as well.
That's fantastic, because I was going to say, if everybody had their own sort of reaction to one or some of the molecules, then that would be very hard, idiosyncratic. But are you finding that there's enough commonality across your participants to say, here's some groups, here's some food types or molecule types?
It's a bit of both. The compound we were targeting is furfural. I've mentioned that one before because it's one of the one that we thought was in coffee that was causing a problem. And out of 30 porosmics, so we've had 15 pre-COVID and 15 post-COVID, and out of 30 of them, something like 24 people found that compound porosmic. 3 of them didn't smell it at all, and the other 3 weren't coffee drinkers or just they found other things porosmic. So for that compound, if you could smell it, most people got it as porosmic, but not everybody. And then I've got another set of compounds which are not based on sulfur, and if they didn't get the sulfur one, most people got this group of other compounds as well, which are nitrogen-based compounds. So it was one or the other, and I've got a pattern coming out where most people react to these compounds, but they also react to other things. So there's a lot of individuality as well. I've got a list of about 30 different compounds, and the ones at the bottom, 1 or 2 people pick up, but there's the ones at the top are picked up by the majority of porosmics. What we haven't started looking at really is personal care, household products, and I think we might get another set of porosmic compounds once we start looking at perfumery and things like that.
Yes, I've certainly heard people talk about household cleaning products in this way, but that's interesting. I want to bring Simon back in because I sort of feel that between the 2 of you, we've got the resources we need to kind of drill down on this. And one of the things you said, Jane, was we smell some of these sulfur compounds that are already in coffee, and at very low concentrations we don't mind them. And then you said, but when the concentration's high, we do mind them. And the thing is, nobody's changed the coffee, so the only thought is we've changed. And I just wondered, and I wanted to invite Simon to speculate and think about what could be going on in us so that we've created the change rather than the chemical mixtures are themselves changed.
Yeah, so just to say, I mean, perfumers have known this for a very long time. The very common compound used in perfumes are things like coumarins or skatol, which is in high concentration smells very fecal, but in low concentrations is actually quite pleasant, and it is used in perfumes. So we've known that there's this variation of again our response over concentration for molecules for a while. So that seems to be a clue to what's going on there, at least partially. To think about what's going on with parosmia, you have to think about what's going on with the normal chemosensation. So we have, in a normal nose, we have a range of olfactory sensory neurons, the nerves that are bearing the olfactory receptors, and each one of those receptors, each one of those neurons has picked one of the 400 different types of receptor genes, and it just expresses that. So each sensory neuron expresses one of about 400 different olfactory sensory receptors. And those receptors they're a mix. Some of them are very broadly tuned. They're promiscuous. They'll react to anything. They'll bind to a whole different set of molecules, and they'll switch on whenever they bind to them. And those signals will then travel up the nerve to the brain and get interpreted in the bulb and beyond. But some of them are very specific. Some of them, as far as we can see, looking at mice and sometimes in humans, they only detect a very small class of molecules or a handful of specific molecules. But when you are smelling a smell, whatever that smell is, you are getting a mixture of molecules going into the nose, distributed maybe by their weight. So they much like Jane's gas chromatography column, they might be separated out a bit as they dissolve into the mucus, the normal mucus in the nose. They may be trafficked by certain proteins that are in that mucus. We're not quite sure about that in humans. And then they interact with the receptors and switch them on. Some of them will activate multiple different receptors, and then you've got a mixture of molecules which are also interacting with multiple different receptors. And it's that pattern of activation that we pull out the sensation of a smell from. So so you get this field of activation across the olfactory part of the nose, and that field of activation has a certain pattern for every single smell. Those patterns of activation are reported up to the olfactory bulb, which is the little bit of the brain just above the nose that's waiting for these signals to come in, and then there's some more processing, and then it goes into the brain where there's more processing and the recognition step and the hooking into the things we're talking about memory, emotion, and instinct. Those kind of things all happen further up. So I think this is where we're kind of settling down to a thesis about parosmia based on Jane's what Jane has reported, that if you diminish this field of activation, if you knock out enough receptors and you only have certain receptors still working, then they give you a salient spike. They get a signal. Let's say the coffee pattern is one of activation of 10 receptors, but the virus or whatever it else is wipes out 9 of them, and you get one that's it's very good at detecting just one part of that coffee mixture, and just gives you that one signal up to the brain. And that is not coffee anymore. Your brain's waiting for the pattern of activation. All it's getting is one single nerve cell, and that then clashes with what you're expecting, and the expectations are abnormal, and that's what gives you distortion. So you're not getting what you're expecting. It gives you the triggered part that we were talking about. It acts in the same way as the normal smell did, so it's rather time limited. But what is more difficult to say is, why is that perceived as unpleasant? Why does it evoke disgust emotion? What is it about single neuron activation or porosity neuron activation, sort of a small group of neurons, that gives you this activation in the absence of anything else? And that I think we still need to think about. I don't know if you have any ideas, Jane. I have some ideas, but they're still in the early stages of being thought through.
I was thinking as you were talking that, sorry, my mind's gone blank. One of the things, dilution of coffee. You can, as normosmics, we can mimic what's going on in parosmia, because in parosmia, all your receptors are dumbed down. And if we just diluted coffee all the way down till we're only perceiving these really potent odorants, we don't perceive them as disgusting. And
Yes, exactly.
we don't get such a spike. So I think there's an amplification in there as well, because we don't get the spike.
Yeah.
It's hard
So
to prove.
I have a rejoinder to that, but yeah, please.
Go on.
So so that only works if the spike molecule, whatever it is only triggering that receptor. If it's triggering some other receptor neurons as well, you can't just isolate the activation of just that one receptor and receptor neuron. And that may be why that. But I mean, you're absolutely right, and that is a central problem. We can try and mimic it as much as possible, but getting that disgust emotion needs to be explained.
This is why I think, now that I'm back, sorry, I disappeared into the ether for a bit. This is why I think it's very interesting to have this interplay between someone looking at the neurobiology of it and someone looking at the chemistry, because we also know that, Jane, that chemical mixtures are very complicated. Odor mixtures are very complicated, and they don't just present their elements and they're all there, but some will suppress others, some will enhance or boost others. So might there be a sense in what we heard from Simon that if some of the receptors are out of play, that it might be that you're just stopping some of the masking or the down-regulating of some of these bad odors?
I think there's something to do with regulation there and feedback mechanisms. And I think that's more for Simon to talk about, actually, because that's where my knowledge stops there. But there's some sort of mechanisms which are certainly boosting the porosmic aromas. I do think there's two things happening when you're recovering as a porosmic. And I think there's one thing where all the other receptors, sensory neurons, are growing back. And that helps to mitigate the porosmia because you're getting contribution from all the others in the field. But I also think the porosmia is being down-regulated as well, because we've had one person who's come back after 6 months and said, my porosmia is so much better. And I said, sit down, we'll test you and see what's going on. And unfortunately, well, it's fortunate, she feels she's recovering, but we saw no evidence of these other receptors being activated. She couldn't smell anything more. Her threshold had gone up, which means that it was worse. So what had happened in her case was that I think she was just down-regulating the porosmia. So I think there's the two aspects, and they've got to both work together. We had somebody who had no anosmia, so she had a perfectly normal olfactory function, scored very similar to me on all the tests, and she still had porosmia. So there's two quite distinct things going on. I think we understand a little bit about the fact that we've still got to grow back all those.
Sorry, I'm just aware that we're over halfway. Some time for questions, unless you want to just discuss a little bit more before we go, and we can continue.
I think unless Simon wanted a quick rejoinder to that, but it'd be nice to have some questions. Remember, we cannot answer medical questions. This isn't a diagnostic setting, and even though we're taking Simon's time, we can't treat him as our sort of local doctor to suddenly advise us. But Simon, did you want to say something about that before we take a question?
Yeah, no personal cases can be discussed, unfortunately. But I mean, this is a medical question, I suppose. No, I think that normosmic porosmic is going to be a real problem for us to think around theoretically, apart from maybe the fact that, you know, the definition of normosmia is, I think, is extremely broad. There are people who have, you know, exquisite senses of smells that you're just not going to capture on a TDI, and so they have headroom to lose a lot of function while still being able to mimic what my nose would do on a standard smell testing.
It's also something,
but it's an important problem to think about.
There's also something to be said here, which I think we should get in, which is people's ability to assess their sense of smell is not very good, usually, for all of us. So, you know, people will say, oh, I've got a lousy sense of smell, I'm not very good. And then you give them the sniffing sticks and you measure them, and they've got very high scores. So this is a puzzle in a way. And I don't know if you've heard, Simon, but I have heard anecdotally from people, well, not even anecdotally, Adam Rutherford, the science journalist, said, having lost his sense of smell, he said, now my sense of smell is amazing, or having had COVID, he said, now my sense of smell is amazing. It's like a superpower, I can smell everything. But I wonder whether it was only when people lost their sense of smell, when it was absent, and they knew what an absence it was, that its return makes them think, wow, the world is technicolor.
Yeah. Yeah, I mean, there's no question. When I started researching the sense of smell and started paying attention to what was, things that I hadn't really paid that much attention to anymore, I suddenly realized what a rich sense it is. And again, this comes back to the same thing, and one of the central, most interesting things about this sense, and one of our biggest hurdles up until now, is that people, I've been saying ever since I started, people don't miss their sense of smell until they don't understand their sense of smell until they lose it. And that's part of our sort of outreach as researchers and as a charity, is about trying to make people appreciate it while they have it.
Yeah.
But you're right. I mean, the evidence is very strong. People are rubbish at telling whether they have a good sense of smell or not.
In both
directions. Very bad.
Yeah,
exactly.
Yeah. So there's a question we have in the chat. Is this Aidan? You were lobbing this up. Diane asks, wondering why there's such wide fluctuations in my parosmia day to day. Progress doesn't seem to be linear. So thoughts about that? Simon first.
That is quite interesting. Again, I mean, we're not sure that the stimuli are the same, and there are a lot of things that modify your sort of baseline. Well, we think baseline smell function. So hormones have an effect, for instance. Hunger has an effect. But yeah, that would be interesting to try and quantify that and to see whether there's a pattern to those kind of things. But yeah, it's not linear. I mean, just to say, I think that's pretty common, certainly in the people I see, that it comes and goes, that it gets worse and better. And this is true for a lot of these sort of sensory disturbances. The same is true for tinnitus. It comes and goes. It tends to get worse when you're stressed and improve when you're not. So I think there's a lot that they have in common there with those kind of disturbances, distortions, in fact.
Just to link on there, Nick asks if there are any typical milestones you see on the path to parosmia recovery. That's quite a difficult question.
Yeah. So I again, so I tend to see people early on and less so after their prolonged recovery phase. But the people that I have talked to who tend to be recovering tend to report it, if anything, retrospectively. So they realize after a while, because of this fluctuating path, that they just look back and think, oh, well, it hasn't been that bad recently, or or it's been sort of decreasing, or I have oh, for the first time my toothpaste doesn't make me want to spit it out. And often actually what improves with the parosmia is, to a certain extent, the perception of distortion, but the disgust element seems to diminish rather than necessarily the altered sensation. So there's some kind of habituation going on where it's less obtrusive and less evoking of primal disgust.
Jane, are there some smells which seem altered to your parosmia participants, but which aren't disgusting? They just know that they're not as they should be.
Oh yes, some people say, yes, that's not disgusting, but it's definitely distorted. It's definitely that aroma. I mean, it's very hard. They say, well, how do I know if it's distorted, because I haven't smelt it before? But they say, yes, it's a parosmia smell, but it's perfectly okay. Yeah. So there are some that aren't disgusting, definitely. Definitely. We didn't measure disgust. We measured, because there's just a limit to the number of things people can assess as these aromas are coming off, and we had no idea how quickly they were going to come off and how many people are going to smell. So we only asked for intensity and whether or not it was parosmic. We didn't go for that extra dimension of valence of disgust,
Mm.
which would have been very interesting, and it would have been more interesting than intensity, to be quite honest, because the intensity figures are all over the place. But that's with hindsight. But there are definitely things that are parosmic but not disgusting, and there are things that are disgusting but not parosmic.
More research is needed.
More research is needed. And other, you were talking about this, and we shot past it but quite quickly, but other sort of food groups that we could roughly group together to say that they're pretty much targets of the things that trigger us, probably because of the molecules they contain.
Yes, I mean, I can answer that one. It is turning out the onion garlic group is a very specific group, and there's very specific sulfur compounds in there that are not in coffee or any of the other. They are specific to that botanical group, so chives and leek and spring onions and all the rest of it come into that group. Then there's the meaty group, which is sort of sulfur compounds, and they're in coffee. And they're in chocolate, and we see a trend there where the sulfur compounds, you don't need a lot of heat to generate these. So a lot of people say that meat cooking is one of the horrible smells. And, you know, these aromas come off meat even at low temperatures. And then there's another group, which is the nitrogen compounds, and you need a lot of heat for those. So those are the deep fried, roasted, toasted, grilled, baked. When people say it's always baked, roasted, toasted, grilled, barbecued, that's nasty, they're probably picking up that group of nitrogen compounds. Whereas people that find meat and coffee and peanuts, they're picking up the sulfur. The coffee is a gem because the coffee has them all.
Yeah.
Coffee, yeah. So we started with the right place. And what we've gone on to do now that we've got more parosmias coming in, we've explored all sorts of different foods now. So we've looked at apples, and we've looked at bananas, and we've looked at wine, and we've looked at crisps and cucumbers, and green peppers is a good one. Green peppers, again, similar compounds to in the coffee, so it's quite interesting, but different compounds as well. So if you're looking at food groups, if you're one of those people that is finds the nitrogen ones particularly offensive, and you'll know that because it's all the roasty, toasty ones, then you have to avoid that sort of food. If you've got an aversion to the sulfury ones, it's meat, it's cooked protein, and that's where the sulfur comes from. Not in the onions, but in the other foods, the sulfur notes come from the protein, they come from the amino acids. So you need to find your proteins that are low in sulfur. Meat's particularly high in sulfur compounds, which is why you get those nice meaty notes. Interestingly, brassica is another group of vegetables, you know, broccoli, cabbage, and Brussels sprouts, etc. And they are very well known for their sulfur compounds too. Partly for taste, but partly for aroma. And it's noticeable that people are not particularly picking up the those cruciferous vegetables as nasty.
That's interesting. So it might be something which has a bit more chemical reaction involved. I mean, I'm wondering about Maillard reaction compounds in cooked meats, in fermented foods, and so on.
Well, Maillard was the ones I was talking about, the nitrogen group, because they are classic Maillard reaction products. You get them when you heat things at high temperature. So roasting, grilling, baking, barbecue. Those are classic Maillard reaction products. And so if you want to eat those sort of products, you just reduce the temperature, reduce the cooking time, try and minimize cooking, and you'll bring those right down. The sulfur compounds are different because they're formed in the Maillard reaction, but they're also formed anyway when you start heating meat up. They're formed much more readily. So cooking at low temperatures is not only not safe when you're cooking meat, but it's
not a good idea. But I was wondering if you have the first sort, to avoid the Maillard, whether poaching a chicken rather than grilling a might help.
Well, I think you get a lot of those aroma anyway. You don't need the high temperatures to get the sulfur compounds. I think you'll still get some of them, and people do report that it's the cooking of the chicken or the meat that's problematic. Once it's well grilled and cooked, that's fine for some people. For other people, it's the grilled and the barbecue, and it's the really high roasty note that they don't like. And it's a balance between all these highly potent aroma compounds. But I found the brassica one interesting because it's got sulfur compounds in it too. That's coffee, but people aren't picking those up.
Even when heated, even when they're heated up?
Yeah. I mean, I've had one person recently, and I'd like her to come back because we didn't have time to do broccoli. But very few people give you broccoli, cauliflower as triggers, as food.
Well, that's good, because people can be eating those good and healthy if they're struggling with the things to eat.
Yeah, I mean, somebody said cauliflower cheese was their go-to because
Excellent. Yeah. Why not? Now we've got a question. I think this is for you, Simon. Are there any specific investigations through MRI going on at the moment, in the light of the fact that we don't have specific treatments? Any diagnostics there?
So nothing at the moment. I think we have talked in Cora about whether or not this would be a fruitful path, not in terms of investigation, but as a research tool, looking at sort of fMRI to see whether we can characterize these reactions, where that disgust emotion is coming from, those kind of things. I was thinking, actually, speaking of diagnostics, whether or not there would be a utility in Jane classifying the parosmia for people and saying, well, you're nitrogen, so you're going to, everyone avoids coffee because coffee's got everything, but you're mainly reacting to nitrogen. These are the kind of things that you should avoid. You're reacting to the high mild reactions, that, stay away from this. Whether there's, most of the time people work it out for themselves sooner or later, but if you can predict that and protect them, or give them things that you think were going to likely trigger them, that might be very useful information, and maybe making sort of characteristic test for those kind of things might be quite useful in the clinic.
Yeah, I mean, sending out a little sample of the compounds would be good. By the way, I just wanted to say, Jane, Kate in the chat said, most green veg are triggers for me, and I say, go and see Jane. Get in touch with Jane.
The thing is about Porosmia and COVID, there's always exceptions. There's always something different. There are no hard and fast rules. So I do know that there are people for which broccoli is a trigger. I do know that. And if you want to come and visit me, I'd love to see what it is that's triggering it. But I suspect it's sulfur compounds that are also present in coffee that are just most people aren't detecting. But yeah.
So we put your email.
Yes, I was just going to say.
Yeah, I've just done that. Yeah.
Olfactory Reading.
Thanks, Aidan.
I have to say, we've kind of come to a pause. I would welcome anybody that doesn't like broccoli, because that would be new, but we've had 15 pre-COVID, we've had 15 post-COVID, we've had 15 non-osmic or non-porosmic people come through. And so we've got a really good base for our coffee. And now we've started exploring other things. So we've started on the garlic and the onion, and we've done that pretty well too, but haven't looked at the data. But now we're looking at different things. So bananas, apples, green peppers, and people that have got those sort of distinct aversions, we'd welcome them to come along, because we've still got gaps. We still don't know enough about those. We've had somebody who doesn't like water. There are a few people who really can't stand the smell of the water as it comes out of the shower, and that's something that's fascinating as well. And I have a feeling that that'll be difficult because, well, for a number of reasons. I think I know what the compounds are, and they're very hard to get on the GCO. But there's these extra things now. These minor triggers are quite interesting for us. We've got the bulk. And we've sort of slowed down, partly because it's a second lockdown. We haven't been stopped, but we've gathered so much data, thanks to you lovely people who've been volunteering, that we really need to sit down, and we're desperately trying to get some papers out so that we can make our mark and say how wonderful our volunteers have been. So that's my aim at the moment. So, I
know both of you are trying to write papers, so we apologise for taking your time this evening. You could be completing one of these.
This is more important.
Tomorrow morning. Yeah.
Jane,
Simon.
Sorry, I was just going to say, if we're talking about bathrooms, should we talk about bathroom smells?
Yes, just what I was coming on to. So I was going to say just that, but coffee is a very useful thing, we've learned, because it's got all the compounds in it that seem to be problematic, and for most people, not everyone, because it's a daily drink and a regular drink, it's a very quick way to pick up that something's gone wrong. But one thing we don't hear about, and it might be a matter of politeness and embarrassment, is that some people have said, and I was talking to a radio interviewer the other day, and she said, funny thing with me was that when I lost my sense of smell and it came back, the baby's nappy, which used to smell terrible, now smells quite nice, she said, and it's okay. And I said, we know a lot about that. This is this funny thing where bathroom smells can suddenly be pleasant and things you like can be unpleasant. The fair is foul, foul is fair. So Jane and Simon, why don't you say a bit more about this?
Well, Jane?
I was going to say, we have actually looked at this in the lab. We haven't put any volunteers through this study. It's just people in the researchers have been through this study. And we can explain half of it. So we've got cases where food smells of poo, to be blunt, and poo smells of food. Now we can sort of explain half of that. We can't explain the whole lot, but we actually put some samples of poo or some poo extracts into our instrument. Now I need to explain, we're a food science department and we do a lot of gut microbiology, and we have people donating poo samples every day of the week, and it's fairly routine for us, and we've got all the handling, the fume cupboards in the category 3 labs, so it was easy for us. And it was actually, I did it as a normosmic or a non-porosmic, and Chrissy did it as a porosmic, and we had one other researcher who did it who was porosmic. So and for the porosmics, it was fine. So we put them on the end of the GCO, and I was, we were getting words coming out like conifer forest and toasted raisin bread and floral and all sorts of wonderful aromas coming out. We were also getting out the coffee, the typical profile of coffee. So all those things that are strong in coffee were coming out, and we had asked for a coffee drinking donor. So, but it was true that the coffee molecules were traveling right the way through. So what the porosmics are smelling is the coffee molecules or the biscuity molecules or whatever it is. Now, for some people it smells biscuity or coffee because they can still smell those. For other people it's sort of distorted coffee. But what porosmics don't get is the really stinky, I mean, Simon mentioned skatol already, but indole, skatol and cresol are the 3 compounds, along with some sulfur compounds, that gives poo its smell. And the 2 porosmics, neither of them smelt any of those compounds at all. So that explains why you're not getting it. Now, why they're not smelling it, we don't know, because they're pretty potent compounds. So there's still a little bit of work to go on there. But when I went on there were tears streaming from my eyes, because those smells were so overpowering that I could barely keep my nose in that nose piece. And, that is what's lacking. That's what porosmics can't pick up at the moment. So that explains, we can explain why the poo smells of coffee or biscuits or whatever, but we still can't really explain why biscuits smell of poo. So that's still that piece in the jigsaw that we're after. But it was an interesting
science. You're doing this for science. It's so impressive.
You than me,
Simon.
Well, I mean, I just are we sure that when the people are saying my biscuits smell of poo, then they're not just trying to evoke that same disgust? I mean, I've kind of drilled into this with quite a few patients when they say, "Oh, it smells of sewage." And you say, "Does it actually smell of sewage? Is that?" And they say, "Well, no, it doesn't smell of anything that I can tell you about, but it makes me feel like that." They use these things in analogy rather than in a precise description of the smell object, and that may be why it's the other way around. That it smells like, it makes me feel like I used to feel when I smell poo. Now I smell when I smell biscuits, and vice versa. And I wonder whether, whether or not, well, I mean, we need to look at the receptors for these things. I personally think, and whether or not there's something about the receptor that predisposes it to being knocked out by viral infection. the one that detects indole or cresol or whatever. I think we know which one detects indole, if I remember correctly. I'll have to go and have a look at that. And whether, because we haven't mentioned it, but one of the things that continues to confound me about this is that we know that the olfactory sensory neurons themselves do not express the ACE2 and TMP RSS proteins on their cell surfaces. They cannot get infected by the COVID-19 virus because it doesn't have the little docking ports to get into the cell. So why certain olfactory sensory neurons should be knocked out and others are left alone is a and common, you know, in it's not just stochastic. It's not just some are and some aren't. There seems to be a pattern to the ones that are involved and the ones that aren't, and that's still, we need to really narrow that down and find out more.
My answer to that is really where this project started, and that I mean, in my mind, they're probably all knocked out, and this is these are the questions that we're asking. It's why we're doing the project. But in my mind, they're all knocked out and they're all growing back slowly. But the ones that are most efficient are the ones that can start to pick up things earlier than everything else. So although we've got a level playing field, the ones that pick up these potent sulfur compounds are essentially efficient and are the first ones to perceive. They need the lowest level of activation to be to cause a trigger.
Yeah.
So I can't see why they would be knocked out in different parts. I mean, it's possible because there are different zones, aren't there, Simon?
Yeah. Well, yes, we think it's certainly in rats. We're not so sure in humans.
Yeah.
But your efficiency thing makes me think. Okay, so you're thinking about the binding efficiency or the recognition efficiency of the receptor, but you'd be able to overcome that with super threshold stimulation. You just give more of something, and it should be able to switch on the other receptor neurons, because that's what the threshold means. But I wonder if there's something else about not necessarily the binding or the recognition step of the receptor, but something maybe about their expression on the cell surface. It's easier to express these. We can make expression systems that force the expression of these receptors on heterologous cells, but they're pretty Frankenstein-y, quite hacky, and you still can't express all the human receptors on cells. There's trafficking proteins and things we don't understand, and I wonder whether it's not the receptor protein per se, but it's the way it gets from the nucleus to the cell membrane. That means that, you know, the ones that detect the peripherals are easier to traffic and therefore get to switch on more easily or are more dense on the cell surface, something like that.
I think that's a question that people have been asking for ages. What makes these compounds so potent?
Yeah.
That's a question we haven't got the answer to. So,
no.
You know.
But here's a crude way of saying something which, if it wasn't for the knocking out of indole and scatol and all the rest, it might have made more sense, but it complicates the picture. Because you might think, in the case of familiar foods smelling odd, that rather like nerve endings that grow back, they can kind of grow back in odd patterns. You know, they can get twisted, and so they just hook up to the wrong ports. And then you think you're getting a recognition that's wrong because you've gone to the wrong address. But then when we go down and look at these bathroom smells, and you're just not even having any recognition, none. It's not even like cross wiring. It's just you're not picking those things out. That complicates the picture. That nice, wrong connections regrowing picture. So the whole thing's still more research needed.
Yeah, that was my working hypothesis until I met Jane, and she just put a torpedo into it. Thanks, Jane. That the miswiring, that was my explanation. But there's several problems with that now.
That does torpedoed, and I know we're coming to the end of our session, but this is also why I do think we need both a chemist and we need the neurobiologist to kind of work together, because what's gone wrong in us and what isn't affecting us in this way, we really need to put those together. So this has been a fascinating session. Before I thank you both, I just want to remind people who've been attending these seminars that there will be another one on April the 14th. I think
November 24th. Yeah. Yeah.
November 24th. Thank you. November 24th, and this is when Nancy Rossen from Monell will be here to talk about, with Frederica Genovese from Monell, to talk about what's known already about the mechanisms of COVID-19 infecting us, so that we have these problems of loss, distortion of smell, phantom smells, and so on. So please do come back for that. But it remains for me to thank Aidan for looking after us, Aidan Kirkwood, for doing the technical assistance. Thank you so much. And also thank you to Jane Parker and Simon Gaine, my colleagues, who I'm relying on to eventually get to the bottom of this mystery. But thank you all, thank you all for joining us, and we hope it's given you
very much
more to think about and some more things that we can learn. So thank you. Good night.
Good night.
Good night.
Thank you.